Okay, welcome my dear friends. This is Dr.PADALA Tirupati, Assistant Professor. Today our eminent resource person, Dr.A.KAVITHA Madam Garu. Madam has joined already. So Naina, shall we start? Naina is here now. Naina Unadama? Okay. So we would like to invite Dr. A.KAVITHA Madam Garu, Assistant Professor, Department of Botany, RBVRR Women's College Autonomous. Madam, please welcome and start your session. Share your screen.
Okay, sir. Okay, sir. Thank you. Good evening to all of you. So this is Dr. A.KAVITHA. I am Assistant Professor from Department of Botany and Food and Nutrition, RBVRR Women's College. So today I would like to go with the topic called as stellar evolution in pteridophytes. So just I'm start sharing my screen. Yeah, I hope you can see the screen over here.
Yes, ma'am. I can see, ma'am.
Can see, right? I'm just putting that in the slideshow. Right. So this is about stellar evolution. So in this we discuss about the different types of steels and its evolution in the pteridophytes. Now, so what we are going to study about here, what do you mean by stellar theory? What are the components of steel? What are the different types of steels that come across in the pteridophytic plants along with their examples, and how the steel is being evolved in these pteridophytic plants. Now, so what do you mean by the steel and what are the components of the steel? So as we discussed in the type studies of Pteridophyta, like in Lycopodium, Equisetum, or in Marsilia, so we discussed that in the anatomy of root, stem, or whatever the leaf also, we have seen a central part of that anatomical tissue or the anatomy. So what is that? It is the central cylinder or the central core of the vascular tissue, which we see in the higher plants. So what are the components of the steel here? The components of the steel include the two main one. One is called as the xylem and another one is the phloem. So next to come surrounding this steel, we can see a single layer of pericycle and we can see the medullary rays or the medulla if present or not. Now, so as we all know, xylem and phloem, these two are considered to be as the vascular tissues of the plant body. That is, they are going to conduct, the xylem is going to conduct the water from the root to the higher plant, that is to the upward parts of the plant body, that is to the stem, branches, leaves, and the tip of the stem and all that. Whereas phloem, it is going to conduct the food material, which was prepared in the leaves. From the leaves, the food material will be supplied to all the parts of the plant body, and the food will be supplied towards downward direction and reaching to the tip of the root and the root parts. So that is the reason why these two are called as conducting tissues, or we are also calling them as the vascular tissues. And this term steel, it was used by the scientist called as Tighem and Dolliard in the year 1886 in the theory of explanation called as stellar theory. Now, what is this stellar theory? What does it what it is? What is the theory proposed by them? According to them, what are the major highlights? Now, steel, it is a real entity and present universally in all higher plants. Yes, stellar region, steel, it is a present in all the higher plants. Whatever the plant you can take, that is the land plants with the vascular nature. Okay, in all the plants you can see, that's why we call it as a universal. In all the plants we can see the presence of stellar region. Now, cortex and steel, these two forms the fundamental parts of the shoot system. The steel and the cortex, they are separated by the endodermis layer. And the main components of the steel are xylem and the phloem. And pericycle, medullary rays, and the pith, they are also the they are also the components of the steel. So these are the highlights of the stellar theory, which were proposed by these two scientists. Now, what are the different types of the steels we observe in the plants, right? From pteridophytes to the higher group of plants. Now, based on the ontogeny and the phylogeny, that is based on their origin and based on their evolution, there are three basic categories of steels were observed in the vascular plants. They are protosteel, siphonosteel, and the solenosteel. So these are the three main categories of the steel. Now, so you can see here what is the difference of lying between each type of the steels here. Now, when we go for the protosteel, so as I explain you here in the equisitum and also in the lycopodium also. Protosteel is such a stellar condition where the steel is without pith region. The steel does not have any pith in the center. Whereas the siphonosteel condition, this is a condition where you can see the stellar region along with it is going to have the pith, but there are no leaf traces or no leaf gaps. And whereas the solenosteely condition, this is the evolved or the advanced type of the steel in which the steel is going to have the pith and along with the leaf gaps or the leaf traces, we call it as. So among this protosteely condition, there are 5 different types of steels. One is called as haplosteely condition, actinosteel, plectosteel, mixed protosteel, and the mixed protosteel with the pith region. This is an exceptional case. Now, haplosteelic condition, this is the condition where the xylem will be in the center and it is surrounded by the phloem. So thus we can see it in example called as rhinia and lycopodium. So these examples are very, very important here. Haplosteelic, protosteelic condition. The example is lycopodium and rhina. In the haplosteel, the xylem is going to be surrounded by the phloem. In actinosteelic condition, the xylem is in the form of a star shape and the phloem will be present on the arms of this star-shaped xylem. So the best example for this actinosteel condition is Lycopodium serratum. And plectosteel, the xylem here will be in the form of plates, and between these xylem plates, we can see the presence of the phloem. So that condition is called as that condition is called plectosteelic condition, and the example is Lycopodium clavitum. Whereas mixed protosteel, here the xylem as well as the phloem, it will be in the form of patches spread all over the stellar region. So that condition is called mixed protosteel. Example is Lycopodium cernum. So if anybody is there here, so please, the students, please, you can note down these examples of protosteel condition, because one question will be definitely asked from this stellar evolution. Okay. So protosteel condition, it is the steel without the pith region. And these are all the different types of the protosteels: haplosteel, actinosteel, plecto, and mixed protosteel. And these are the examples. And the mixed protosteel here, it's an exceptional case, I told you, with the pith region. And example is hymenophyllum. And the second type of category of the steel, that we call it as siphonosteel. So as I told you here, the steel here is without any pith region in the center and there are no leaf gaps. So when we go for this is again divided into 3 categories here. One is called as clado-siphosonic siphonosteel. That is here, there is no pith region and there is no leaf gap. Example is Selaginella. And the ectofloic siphonosteel, the name itself implies ecto means outside, floic means related to phloem. So the phloem will be outside and the xylem will be in the center. So like that, you need to remember. Siphonostele is the one where the stele is without any pith region, with pith region. The pith will be in the center, and surrounding the pith will be the row of xylem vascular bundle, and surrounding it will be the phloem. So that's the reason why we call it as ectoploic siphonostele. Amphiploic, amphi, that means in between. That means phloem will be lying in between the two xylem, that is primary xylem and the secondary xylem. In between these two, or we call it as the outer xylem and the inner xylem. In between these two xylem elements, we can see the presence of the phloem. That's we call it as the example, we call it as the Marsilia rhizome, amphiploic siphonostele. Now, the next third category of the stele, that is called as solenostele. So here the stele is with pith region as well as the leaf gap. So in this, there are different types of solenostelic conditions. One is called as ectoploic. So just now I told you, phloem will be external to xylem with the central pith region and along with the leaf gap. Next, amphiploic. The phloem will be on both the sides of the xylem. Next is dictyostele. Here we can see the presence of many meristeles. That is the primary stele-like conditions we can see. Next, polystelic condition. Many number of vascular bundles, or in these vascular bundles are in the form of many circles. So example here is pteridium. Dictyostele example is teres. And eustelic condition. So this is the condition where the vascular bundles are arranged as a broken ring. That is, like on different radiating arms, we can see the presence of the vascular bundles. And atactostele, so these are the one which what we can see in the higher plant, that is dicots and the monocots. In the dicot stem, the vascular bundles are in the solenostelic condition with the eustele, and atactostele in the monocot, that is the scattered arrangement of the vascular bundles. So that condition is called atactostele. So this is what I explained just now. Protostele. Pith is going to be absent. In the center, we can see a solid core of the xylem. And it is the simplest stellar organization. And this is considered to be the most primitive one among the plants. Next, I told you this is divided into 5 types: haplostelic, actinostele, plecto, mixed, and mixed protostele with the pith. So this is a haplostele condition, with example is the Selaginella. So you can see this is a Selaginella stem. So you can see here, this central part here, this is the xylem. A central part is the xylem, and it is going to be surrounded by the phloem, uniform layers of the phloem. And also you can see the pericycle surrounding the xylem and the phloem. It is also, this haplostelic is the most primitive one, and it is oftenly observed in the fossil genera and also in the Selaginella and the some species of the Lycopodium. Okay, this is phloem, this central part is right. This is haplostele. Next comes to the actinostelic condition. So protostele here is with the central xylem, and the xylem is in the form of a star shape. You can look into this here. This is a star-shaped xylem, and the phloem will be at the arms. Okay, so here on the arms of the xylem, you can see here the presence of the phloem. Next, it is represented another category of the protostelic condition, that is plectostele. So this is an example of Lycopodium clavitum. Lycopodium. So you can see here, these all are the plates of the xylem. And in between these plates, we can see the phloem patches. And it was named by the scientist called as Zimmermann. And the example of plectostelic condition observed in Lycopodium clavitum. Next, this is a mixed protostele. Example is Lycopodium cernum. So here, both xylem as well as the phloem, they are in the form of the patches. So this is about the advanced type of the protostele, which is called as protostele with the pith. So we are studying here the evolution. So that is the reason why here the primitive one of the steels are the protosteel, and that too, that is the haplosteel. That is a primitive one without any pith region. But as we move on to the advancement of the stellar evolution, so among this protostelic condition, the mixed protosteel here with the pith region, it becomes the advanced. So the next evolution of from this protosteel will be the siphonosteel. Okay, so like that we need to remember. So here this is the advanced type of the mixed protosteel with the pith region in the center, and the xylem and the phloem here, it will be in the form of the patches. Xylem is alternating with the phloem and they are scattered throughout the stellar region. Example here it is hymenophyllum demisum or lepidodendron. So the another category we discussed just now, that is siphonosteel. So what is the steel? This is a steel with the pith at the center region, and the pith is going to be surrounded by the xylem. So here the siphonosteel is the advanced form than compared to the protosteel. Now, so this siphonosteel again, it was derived or it was originated from the protosteel by the formation of the pith, and centrally we can see the xylem core, and it is replaced by the parenchyma here in the center. So these are the two theories which were proposed by the scientists to explain about how this siphonosteel was originated from the protosteel. So first theory, it was called as interstellar origin, whereas the second theory is called as extrastellar origin. Now this interstellar origin, it was proposed by the scientist called as Bower and supported by Fawn. So according to this theory, the innermost vascular tissue in the protosteel, it changes into the parenchymatous cells with a pith development. Whereas extrastellar origin, it was proposed by Jeffrey. So what he says that pith and the cortex, they are the homogeneous structures, and the pith is formed by the invasion of the inner cortical parenchymatous cells into the steel. That is the inner cortex, which is there in the cortical region. Some of the cells of these cortical cells, they will invade into the stellar region. And that too, they are of the parenchymatous type. So that is the reason why the pith was formed in the center. And invasion of the pith occurs through the leaf gaps or the branch gaps. Now, when you talk about here, there are two different types of siphonosteels. One is called as ectofloic siphonosteel, and another one is the amphifloic. So these two types of siphonosteels, they were based on the position and the distribution of the phloem. Now here, this is the ectofloic. So already I told you, ecto means outside. That is, phloem is going to be C here. So you can see here, this is the phloem. The phloem will be present on the external side of the xylem. So this blue color one, what you can see in the diagram, that is a xylem. And this is a central pith region. And outside to the xylem, here it is a phloem. Okay. And this phloem is externally surrounded by a single layer of endodermis as well as the pericycle. And here, the leaf traces are present. These are the leaf traces in the cortical region, but there are no leaf gaps here. So example is osmunda. So very good example here. So this is a central pith region. It is going to surrounded by, this is a larger region here. What you can see, that is a xylem, and outward to that is a phloem. Okay, that the condition what we called as ectofloic siphonosteel. Amphifloic, that means phloem will be on both the sides of the xylem. So you can see here, the blue color one is a xylem, and on either side, that is, this is the outer phloem and this is the inner phloem. Okay, so it is a very good example here. We can see in the Marsilia rhizome. I told you this point also. Amphifloic siphonosteel, and here the central one is the pith region. So you can see here, this is a central pith region, and here you can see a round of this is a xylem, and here this part below the xylem, this is one phloem ring, and outside the xylem, that is another phloem ring. And one more important point here to discuss here is we can see the presence of even endodermis and pericycle. They are also distributed. The endodermis and the pericycle, which is surrounding the pith, we call it as inner pericycle and inner endodermis. And the one which is surrounding the outer phloem, that is also one pericycle and one, that is the outer pericycle and the outer endodermis. So that point has to be noted. In Marsilia rhizome, we can see that structure called as amphiploic siphonostele. So that is about the ectoploic condition of siphonostele and amphiploic condition of the siphonostele. Example, this is Marsilia rhizome and ectoploic is the osmunda. Next, solenostele. So this is the stele, what I told you, which is the advanced form of all the stele categories. So here we can see the presence of the pith region in the center and with the leaf gaps. So such a condition is called solenostelic condition. So there again, these solenosteles are divided into 6 different types based on the plant kingdom: ectoploic solenostele, amphiploic, dictyostele, leustele, and atactostele. So you can see here, this is an ectoploic solenostele. Ectoploic, just now how what I told you. The phloem will be towards external position of the xylem. So this is a xylem, blue color one, and this is a phloem surrounded by pericycle and the endodermis. And what is the difference here? Central pith region you can see. Now what is the difference here between siphonostele and the solenostele? In the siphonostele, we can't see the presence of any leaf gap here or the leaf trace. In some example, we can see the leaf trace, but no leaf gap. But solenostele condition is such a stele where we can see the formation of a leaf gap here. Okay, that is the only difference lying between the siphonostele and the solenostele. So this is the ectoploic where phloem is outside. Inner to that will be the xylem, central pith, and continuity of that will be the leaf gap. That is ectoploic. Amphiploic. So you can see here, this is a gap here. This is a leaf gap you can see here. So this is the central pith region and the central, this will is the xylem, and on both the sides we can see the presence of the phloem surrounding the pericycle and the endodermis. Example here it is adiantum. Next, dictyostelic condition. So this is the condition, best example is a terrace. So this is the condition where the solenostele will be broken into separate vascular bundles. So you can see here, this is one vascular bundle, another one, this. So all the vascular bundles, whatever are there, they are observed in the form of broken network. Okay, and this is due to why that gap here, why there are why there is separation of these vascular bundles here is due to the presence of leaf gaps. So these are the leaf gaps you can see here. So there are many leaf gaps here due to which the vascular bundles are getting separated or they are broken down into separate vascular strands. So the best example here, this condition is called as dictyostelic condition. So each one here represent one stele. Okay, stele and enti. Stele means it is having endodermis, pericycle, and vascular bundles. So here you can observe this part. Okay, endodermis is there, pericycle is there, and these are the vascular bundles. There is a leaf gap. This is another vascular strand, we call it as. This is another stele. This is another one. Okay, so each one here represent a stele. So that is called as meristele. Small vascular strands. Okay, with endodermis and with pericycle, with separate endodermis and pericycle, we call it as meristele. So we can see here the presence of many meristeles here. So such condition is called dictyostele. An example here is adiantum, the species called as capillus, and another genera is terrace. Next, polycyclic stele condition, that is solenostele polycyclic. selenosteel, and polycyclic dictyosteel. Polycyclic means presence of many vascular bundles. So then it is called as eustelic condition, but it is a modified of ectofluic siphonousteel. That is eustele condition. So which we can see here in the dicot stems generally. Okay. In the anomalous secondary growth, we can see the vascular bundles. They will be in different, there will be in ring. They will be arranged in the form of different rings. So this is one meristele, this is another meristeles, like that. Even cortex also consists of the steels. So such a type of stellar condition is called as eustelic condition. So this can be observed in the higher plant, that is the dicot stems. Atactostele here, that means to say the vascular bundles we can see, they are scattered in the ground tissue. Especially the monocot stems, when you take the transfer section, we can see there are many vascular bundles which are scattered in the ground tissue. So such type of stele is called atactostele. Example is the monocot stem. I hope this is clear for you all. So this is about the different types of steles and how they are being evolved. Can just stop sharing here. Any doubts regarding this particular topic, please. So here this topic is a very, very important topic. Very, very important. So examples, what I quoted are definitely at least one question will be asked from this point, from the examples in your competitive exam.
Ma'am,
Yeah. Yes.
ma'am, in stellar evolution in pteridophytes,
Yeah.
you
Yeah, you
explained three
have.
types, no, ma'am.
Yeah, you are asking about important questions from this topic, right?
Yes, ma'am.
Right. So what I told you just now. So all the examples what I quoted. Like for example, if it is a haplostele condition, okay, example of haplostele, they will ask you the examples like if it is observed in Lycopodium which species, or they will ask you Lycopodium, Terris, Adiantum, like 4 genera they'll give, and you have to choose the correct answer from that. Haplostele condition, what I told you, haplostele condition is a primitive one. Primitive of the protostele condition, which was observed in the fossils, Rhynia, or in the basic of Lycopodium. So if Lycopodium is there, you can take that multiple choice of Lycopodium. Like that. Suppose if it is plectostele condition. Plectostele means what? The vascular bundles are in the form of plates. In which genus? Dash, or the plectostele condition is observed in dash. So they'll give you Lycopodium or 4 different species of Lycopodium. Lycopodium cernum is there, Lycopodium clavatum is there, Lycopodium phlegmaria is there. So in which one this plectostele condition is observed? So that species you need to tick. Okay. So please just focus on this stellar evolution with examples. Make a note like plectostele example. Mainly they focus on examples only. Okay. So please make a note of that and you prepare.
Ma'am.
I hope this is clear. Yes, ma'am.
Ma'am, this stellar evolution is having already in MSc Botany in MCQ's book. There no, ma'am.
Yeah.
In that book, I already read, ma'am. But
yeah,
these 3 types is not there in that book, ma'am.
Yeah, that's what we don't know. All the books they won't focus on all these points what I explained, but the paper setter definitely will focus on all these types. Okay, just make a note of it and you can prepare. Okay, already this you studied in your degree, right? In degree you have studied all these.
Yes, ma'am.
Definitely the questions will be asked.
At least 1 question, 2 questions I will give, ma'am.
Yeah. One definitely, and 2 exceptional. We can't say that, but 1 is definite. So sir has already posted the previous papers. Please, you can go through those papers and you can read accordingly. Okay. Right. Any more doubts here regarding this?
Ma'am, give at least one question or two questions, ma'am, or otherwise more than two questions, ma'am.
No, not more than two, one or two. That's it. That makes a big difference for your rank.
Okay, ma'am.
Right. If you are
But
1 mark lesser than the another one, like if you get 1 mark less than your other people, your rank will be in hundreds. Your rank difference will be in hundreds. Okay. So you should not
yes, ma'am.
miss each and
But
every point.
yes, ma'am. But they will give easy question or hard question, ma'am.
It depends on paper setter. How can I say? I'm not setting the paper, right? It
Naina,
depends on the paper.
Naina, I have already sent in the WhatsApp group all the previous papers explanation. Completely analyzed paper I have sent to you. So whenever you have free time, please listen carefully and question how the questions are framed. Which type of questions has given in our previous papers. I have already sent you each and every paper analyzed. 4 options I have analyzed. So don't worry. And also last year paper I will send within 2 days. I am already analyzed that paper, sent within 2 days. Okay, don't worry.
Okay, sir.
Madam, really
Yes, sir.
wonderful session, madam. Thank you very much. So
Okay,
tomorrow
sir. Thank
onwards,
you.
madam is going to explain about the new topic, that is gymnosperms. Okay. So thank you very much, madam.
Yeah.
I will
Thank
wind
you.
up the session
Thank you
with
for giving me
with
the
your
opportunity,
permission.
sir. Thank you.
Dr.A.KAVITHA madam garu, Dr. And other participants, thank you very much. Tomorrow we will meet same time. And one more important notice, 7 to 8, Dr. D. Nagaraju sir will going to start new topic, that is mycology, fungi. Okay. So join sharp 7 p.m. Okay. Thank you very much, madam.